Hello students, this is Mrs. Shweta Laddha from Fergusson. We had started with our first chapter, Reproduction in Lower and Higher Plants. And we have already finished with pollination, types of pollination, contrivances, that is adaptation for self-pollination, cross-pollination. And then we had discussed about the benefits, advantages of both self-pollination and cross-pollination. Today we are going to start with actual process of reproduction which occurs in angiospermic plants. Angiospermic plants are flowering plants. Now like other plants, they are also having two generations. They also show alternation of generation. The sporophytic generation is the dominant. You should know students, sporophytic generation is always diploid. Okay, the green plants which you see, the mango tree, the neem tree, the hibiscus tree, the rose tree, the trees that you see, that is the sporophytic generation which is diploid in nature. On this sporophytic generation, the gametophyte arise. The gametophyte is nothing but the flower. So you have already studied flower, structure of flower last year. I'll just take a brief revision about structure of flower. So flower, the stalk of the flower, it is called as pedicel. The end of the pedicel, it is swollen part, it is called as thalamus. Actually, it is a condensed stem only. It is having nodes, internodes, and on it arise the different floral whorls. There are 4 floral whorls, out of which 2 are essential, 2 are accessory. The essential whorls are androecium, the male reproductive whorl, and the gynoecium, the female reproductive whorl. How they will participate, we will see this part in our presentation. So let us begin with our PowerPoint presentation. Look at this diagram. The stalk of the flower, it is called as pedicel. On the top of the pedicel, this expanded portion which is called as receptacle, it is also called as thalamus. On this arises the calyx. Calyx is the accessory whorl. The members of calyx are called as sepals. It protects the flower in bud condition. It is green in color, so it carries out photosynthesis. The next floral whorl is corolla. It is colored. Members are called as petals. They attract insect for pollination. We are interested about the essential whorls. The first one, androecium, and then the gynoecium. Androecium, the members of the androecium, they are called as stamens. If you look at the stamen, it has 3 parts, the anther, the connective, and the filament. Okay, this is the anther, this part which connects is connective, and this is the filament. Of course, anther produces pollen grains, and pollen grains will give rise to the male gametes. This portion is the female reproductive whorl, which is called as the gynoecium. Members of gynoecium are called as carpel. They are also called as pistil. Each carpel or pistil consists of stigma, the upper whorl, upper sticky part, which is stigma. The pipe-like portion, it is called as style, and this swollen portion, it is called as ovary. Inside the ovary, the ovule arises, and inside it, this ovule, after fertilization, it will give rise to the formation of seed, and the wall of the ovary, it forms the fruit. So basically, gynoecium and androecium are the reproductive whorls. So let us study in detail how this reproduction occurs. Angiospermic plants, I told you sporophytic is the diploid and gametophytic is the haploid generation. Gametophytic generation produces flower. Flower produces male gametes which are haploid, which will be produced in the anther in the form of pollen grains. Pollen grains will give rise to the male gametes. Female gametes will be produced by gynoecium, and they are also haploid, and finally they will produce egg. Fertilization between the male gametes produced by the pollen grains and the egg, it will result in the formation of zygote. Zygote is diploid in nature. This will then undergo embryogenesis, and again it will give rise to sporophytic generation. Sporophytic generation is dominant. Gametophytic generation is for short time. Now let us begin with structure of anther. So before that, let us see what is a stamen. Every stamen, the member of androecium, it consists of anthers, connective, and filament. This portion is connective and filament. So members of androecium are stamen. Each stamen consists of long stalk. And what is the long stalk called? It is called as filament. The upper broad knob-like bilobed anther is connected by a connective. Number of stamens, they varies in different species. Let us see the structure of anther. a normal bithecous, that is dithecous anther, it is made up of two anther lobes, 1 and 2. Such type of anther lobes, if there are 2 in number, we call it as dithecous or bithecous, which are connected by a sterile connective. So they are connected to each other by a sterile connective. 2 anther lobes contain 4 elongated pollen sac. Look at this picture. You can see 4 pollen sac and they are elongated. Isn't it? Even it is taken a cross section. Just imagine they run from ear till ear. So there are 4 elongated pollen sac. As there are 4 in number, what do we call it? We call it as tetralocular or tetra. Tetra means 4. Sporangiate because they are going to form the microspores. Also called as microsporangia. Remember these words, very important words. Bithecous, dithecous anther, tetralocular or tetrasporangiate, microsporangia, pollen sacs. In simple word, microsporangia are also called as pollen sacs. You can see here the labeling, pollen sac. What is their function? They produce pollen grains. Process of formation of microspores, that is pollen grains, in the microsporangia or pollen sac, it is called as microsporogenesis. Students, this question can be asked for 1 mark. What is microsporogenesis? Simple word. What are microspores? Pollen grains. Where are they formed? In the microsporangia. So process of formation of pollen grains in the pollen sac or microspores in the microsporangia, it is called as microsporogenesis. So this is an important definition. I hope you understood what are pollen grains. What is the other name for pollen grains? Microspores. Where are they produced? They are produced in pollen sac. What is the other word for pollen sac? Microsporangia. How many chambers are there? How many microsporangia are there in normal anther, bithecous anther? 4. So it is called as tetralocular or tetrasporangiate. I know words are difficult, but you understand the meaning and try to remember these words. Let us see the structure of a pollen sac. What do we call scientific term to pollen sac? Microsporangium. I will go slowly one by one. Let us see. Anther wall, it consists of 2 regions. Okay. The first region, it is called as epidermis. You can see in the picture, it is shown by black color. Now I am making it red. This is epidermis. This epidermis, it is single layer. Then the second layer is archisporium. This archisporium, you cannot see any archisporium labeling in the diagram because archisporium is made up of outer parietal layer. Now look if you can see the outer parietal cells and inner sporogenous tissue. So it consists of outer parietal layer and inner sporogenous tissue, archisporium. So what are the 2 layers present in the anther wall? Epidermis. How many layered? Single layered. Second is archisporium, which is made up of parietal layer and sporogenous tissue. Now, so anther wall, I have made a brief chart for you. This will really help you to remember. You also draw this chart with me. Anther wall consists of 2 layers. What is the outermost layer called as? Epidermis. How many layered? Single layered. And the second is archisporium. Archisporium is made up of parietal tissue and sporogenous tissue. Correct? Now this parietal tissue is further made up of 3 layers. Endothecium, middle layer and tapetum. See, I am repeating. Anther wall made up of epidermis, single layer, archisporium, multi-layered. Archisporium is made up of parietal tissue, sporogenous tissue. Parietal tissue is then made up of endothecium, middle layer and tapetum. Every layer has its own function. And the sporogenous tissue, it will give rise to the spore mother cell, and spore mother cell are actually going to undergo meiosis and produce microspores. Okay, let us see in detail now. First, we'll discuss about epidermis. So epidermis, look at this picture. It is the outermost layer. See this outermost layer, I am making it dark red. This outermost single layer, it is epidermis. And the cells are flat. Look at the structure of the cell. They are flat and they are stretched. They are stretched because they are covering the entire pollen sac. And of course, if they are outermost, what will be their function? The function will be protective in nature. It protects the anther. So epidermis, outermost layer, single layered, stretched cells, and it is protecting the anther, greatly stretched. Now let us see again. So we saw anther wall made up of epidermis, single layer, stretched cell. Let us start with now Archisporium. Brief revision of Archisporium made up of parietal tissue and sporogenous tissue. Parietal tissue made up of endothecium, middle layer and tapetum. Now we are going to study the discussion about parietal tissue, and in parietal tissue we'll see what is endothecium, what is its role. So let us see outer parietal layer. Outer parietal layer, the first layer in outer parietal layer is endothecium. Look at the diagram, endothecium. Next to the epidermis lies endothecium, just beneath the epidermis. Cells are radially elongated along the radius, and they're elongated and they have fibrous covering, thickened covering, which is made up of a carbohydrate callose. This is important, can be asked for one line or MCQ. The endothecium cells, they are thickened because of deposition of because of deposition of callose. They are hygroscopic in nature. Hygroscopic means what? They are water loving. They absorb the moisture, and therefore they play a very important role in dehiscence of anther. Can be asked, what is the function of callose or what is the function of endothecium? Endothecium are the layer of cell present just below the epidermis. It is the outermost layer of the parietal cell. The thickening is of callose, which is hygroscopic in nature. It absorbs water and it helps in dehiscence. Dehiscence means breaking of the anther to release the microspores. Cells located in the shallow groove between the two microsporangia remain thin-walled. Where? This region. This is the shallow groove. Groove means Marathit आपण खाच म्हणतो त्याला. So this is the shallow groove. Here in the shallow groove, the two microsporangia, the cells of this region of endothecium, they remain very, very thin. They are thin. So as soon as they absorb water, what will happen? It will absorb water. Because they are thin, there will be a lot of pressure and it will break. And if this breaks, what happens? All these microspores will be released outside. So this is representing the line of dehiscence. Dehiscence, simple meaning breaking, rupturing. Now let us see the next layer. The next layer just below the endothecium is middle layer. It is 2 to 3 layered. So epidermis single layer, endothecium single layer made up of callose thickening, helping in dehiscence. Next to it lies the middle layers. Look at these middle layers, 2 to 3 in number, made up of parenchymatous cells. They are flattened and thin-walled. These cells are flat and thin-walled. They degenerate at maturity. They break down at maturity, and two pollen sac will merge to form one chamber. So this pollen sac and this pollen sac, they will merge, they will join together because of the rupture of the middle layer, and it forms one chamber. Inner to it lies tapetum, most important and interesting layer, tapetum. It is innermost layer, single layered. Cells are large in size and they have diploid or polyploid nucleus. Very important, they are having a diploid nucleus, tapetum, or they are having polyploid. What do you mean by polyploid? Diploid means two set of chromosomes. Polyploid, many set of chromosomes are present in the nucleus of the tapetum. And they are surrounded by dense cytoplasm. Very important function. They are going to provide nourishment to the sporogenous tissue. One mark question. What is the function of tapetum, or who provides nourishment to the sporogenous tissue? Tapetum provides nourishment to the sporogenous tissue. It also contributes in the formation of sporopollenin. Very important component of pollen grain. Pollen grain, the XII, the outer wall of the pollen grain, it is having this sporopollenin, and because of sporopollenin, the pollen grains are able to stay in nature for hundreds of years without getting decomposed. So sporopollenin is a very important component which is formed by whom? It is formed by tapetum. These two important functions of tapetum. They are providing nourishment to the sporogenous tissue. They contribute in the formation of sporopollenin, and they are polyploid or diploid in nature. Now we will discuss about the pollen sac. Just 1 minute, I'll tell you the tapetum. So this is the tapetum. You can see they are elongated, they are triangular, diploid, polyploid, and they are covering the entire sporogenous tissue because they are going to provide nourishment to this sporogenous tissue which is present inside. And they are also going to add sporopollenin to the XII of the pollen grain. Now we are going to study about this pollen sac or Microsporangium. In an immature anther, inner to the tapetum, microsporangium, a compact mass of diploid sporogenous tissue is present. This diploid mass, it is mass of sporogenous tissue which is diploid in nature. Cells of these tissue undergo mitosis and or they may directly function as microspore mother cells. You can see in the picture microspore mother cells. From where are they derived? They are derived from sporogenous tissue. By which division? Mitotic division, or directly the sporogenous tissue can act as the microspore mother cell. These are very important. Microspore mother cell, MMC, you can write MMC means what? Microspore mother cell. They undergo meiosis. So when they undergo meiosis, they give rise to 4 haploid microspores. This is very, very, very important. One microspore mother cell give rise to 4 haploid microspores. Sometimes they ask you question, how many seeds will be formed? I'll take one example at the end, but you remember this. One microspore mother cell, because of meiotic division, give rise to 4 haploid microspores. Young microspores are present in the form of tetrahedral tetrad. What is the meaning of the word tetrahedral tetrad? Look at this picture. This position, this type of arrangement of the cell, they are 4 in number, but they are arranged in such pattern. It is called as tetrahedral tetrad. This process of formation of microspore is called as microsporogenesis. I think now it has become very easy for you. Quick revision, brief revision. Outermost layer, epidermis. Next layer, it is consisting of layer which is next to the epidermis, endothecium. Next to it, middle layer made up of 2 to 3 layers of parenchymatous cell. Next layer, tapetum, providing nourishment, adding sporopollenin to the axon of the pollen grain and nourishment to the sporogenous tissue. This sporogenous tissue, it is diploid in nature. It undergo mitotic division, give rise to microspore mother cell, MMC, which undergoes meiotic division to produce 4 haploid microspores. This process of formation of microspores is called as microsporogenesis. Students, this is very important and I think it is very easy now. Good diagrams are given in textbook also. You can revise it from textbook once again. And I hope you are taking down the notes when I am teaching. That will help you to make charts, to make notes when I am teaching. Look at this picture again, I'll show you. So this is the entire anther. It is a TS of dehisced anther. Here you can see the middle layer, it has been what we will say degenerated, and so the 2 pollen sac is become now 1. And this is the region which is called as a line of dehiscence, the stomium, and through which all the pollen grains, the microspores are released outside. So now we will study the structure of pollen grain. Okay, let us begin now and start. What is the structure of pollen grain and how this development of male gametophyte occurs. So if you look at the structure of the pollen grain, a diploid microspore mother cell, just now we saw microspore mother cell, diploid in nature, undergo meiotic division to give rise to haploid. Kiti bantat? 4. 4 haploid microspore. And what is that process called as? Microsporogenesis. Every pollen grain, single pollen grain is unicellular. Look at the diagram, it is single celled. Uninucleate, look at the diagram, there is a single nucleus in the center. It is spherical or oval or ellipsoidal or triangular in shape. Look at the structure, it is circular, sometimes it is oval, triangular structure. The cytoplasm is surrounded by 2 layers, outer exine and inner intine. Look at this, this is the cytoplasm. It's not labeled, but this is cytoplasm. And this cytoplasm, it is surrounded by 2 layers. Inner layer is called as labeling. This inner layer which is white in color, it is intine. Now I am making it red. This is intine and outer is exine. One more thing you might have noticed, exine is not continuous but intine is continuous. Exine is cuticularized and it is made up of fatty substance called as sporopollenin. This sporopollenin, it is a fatty substance. And who secreted this sporopollenin? The tapetum. It secreted the sporopollenin. Intine is made up of cellulose and pectin. The intine, it is made up of cellulose and pectin. Again very important, can be asked for 2 marks question. Draw a neat label diagram of pollen grain or explain the structure of a microspore. So that time you should remember unicellular, uninucleate, Then you can write down the shape. It is spherical, ellipsoidal, oval, vagare. Then there are two layers, exine, intine. How is exine? Discontinuous, cuticularized, made up of fatty substance, sporopollenin. How is intine? It is continuous and it is made up of cellulose and pectin. Exine is thin at few places, and these places where the exine is thin or almost absent, it is termed as germ pores. How many germ pores can you see in this diagram? 1, 2, 3. So when there are 3 germ pores, it belongs to a dicot spore. But a monocot spore is uniporate. So uniporate spores are present, pollen grains are present in monocot, but triporate are present in dicot. This is again very important, can be asked for MCQ. What type of pollen grain are present in dicot? So they are triporate, and in monocot they are uniporate. They have a single germ pore. And there is a very big role of these germ pores because they are going to help in the formation of pollen tube. So I hope you have understood the structure, and look at the different shapes of the pollen grain. They are of different shapes. They are beautiful actually if you see them under microscope. Some are round, some are oval, some are triangular. Some you can see there are spiny. If you remember, we had studied during adaptation, spiny pollen grain is present in insect pollinated plants, and smooth walled is present in wind pollinated or anemophilous. So those points are also important, and see how they are interconnected. So this was about the structure of pollen grain. Pollen grains, more details about pollen grains. The branch of study of pollen grain, it is called as palynology. The process where the branch of biology where we study entire detailed things about pollen grain, it is called as palynology. In Calotropis, I had taught you this also in Hercogamy and orchids, pollen grains of each anther, they are stored in mass, and this mass, it is called as pollinia. And it is a very good example of Hercogamy found in milkweed. Milkweed is Calotropis, and therefore self-pollination is prevented and cross-pollination takes place. Pollen grains may cause allergies in few people. You might have heard pollen grains allergy ahe. Pollen tablets sometimes are used as food supplements because they are rich source of nutrition, especially by athletes. So as they have different types of vitamins and minerals, they are a good source of nutrition. Now we will see the development of male gametophyte before pollination in the pollen sac. So if you remember our pollen grain, it was uninucleate, oval, surrounded by granular cytoplasm. Inner layer was called as intine, outer is called as exine. Exine is discontinuous. So this was exine. When in situ, when it is still in the pollen sac, so remember, it is still inside the microsporangium, it is still inside the pollen sac. The protoplast of the pollen grain divides mitotically to form 2 unequal cells. Look at this cell. This is one cell, and this other remaining larger cell is the second cell. So when it is still inside the pollen sac, it forms 2 cell structure. The smaller one is called as the generative cell, and the larger cell, it is called as the vegetative cell, or it is also called as the tube cell. Okay. So smaller cell is generative cell. You can see in the picture the smaller cell is generative cell, and it is having a large nucleus as compared to the size, whereas the larger cell is called as the vegetative cell or tube cell, and it is having smaller nucleus. It has lot of reserve food. You can see so many vacuoles are present. Why will be the vacuoles present? To store the food. I repeat again, if you are not following this diagram, let me draw this diagram again for you so that you understand. So this pollen grain, when it is still in the pollen sac, it undergoes mitotic division and it gives rise to a 2-cell stage. So initially it was uninucleate, exine is there, intine is there, and triporate. What happens to this? Undergoes mitotic division. One gives rise to a smaller cell and other is the larger cell. What is the smaller cell called as? The generative cell. What is the larger cell called as? Vegetative or tube cell. And vegetative or tube cell, it consists of a smaller nucleus, but the generative cell, it consists of a larger nucleus. Vegetative cell has lot of vacuoles. 2-cell stage is formed. The larger cell, as we know, vegetative cell or tube cell, having large vacuoles, lot of cytoplasm and lot of reserve food. Generative cell lacks definite cell wall. You can see in the picture, it was shown dotted because it is not having a definite cell wall. and it is suspended inside the cytoplasm of vegetative cell. It is present inside the cytoplasm of the larger tube cell or vegetative cell. In most of the angiosperms, pollen grains are released in the 2-cell stage. So, pollen grains usually release during this phase, during dehiscence. Partially, sometimes this structure is called as a partially developed male gametophyte. It is not yet completely developed, but very, very rare times, generative cell again divides mitotically and 2 male gametes are also formed. So, it sometimes releases in 3-cell stage, but usually it present in 2-cell stage and it is released during 2-cell stage. Now this pollen grain goes and falls on the stigma of the flower of the carpel, and let us see what happens on the stigma. So when it deposits on the stigma, the 2-cell pollen grain is deposited on the stigma. There is lot of sugary substance on the stigma. It will absorb the sugary substance and it will increase in its volume. It creates a pressure on the intine, and so a pollen tube. Initially, pollen tube will be emerging from all the 3 sides, but finally one pollen tube is successful. One pollen tube formation occurs. Intine of the pollen grain emerges, and this pollen tube, it starts traveling towards the style. The pollen tube have 2 haploid, very important, listen carefully, non-motile male gametes, which are formed by mitosis from the generative cell. The generative cell form these sperm cells, which are also called as the male gametes, and the tube nucleus is also present. And this pollen tube then will, suppose this is the style, so it will start traveling inside. After germination, the pollen tube starts traveling inside to reach the ovule because of the chemotactic movement, which is having 3 nucleus, 2 of the male gametes and 1 is of the tube cell. This movement is due to chemical movement. Sterile vegetative nucleus will degenerate, and this represents the final development of the male gametophyte. So students, just a brief revision. This is the stigma, this is the style, this is the ovary. What is there in the ovary? Ovule is there, right? Now what happens? The pollen grain, it falls over here. When pollen grain is falling on the stigma, it is 2-cell stage. The 2-cell stage is having the smaller one, which is the generative cell. The larger one is the vegetative cell. Now this will start growing because of the pressure. It will form on the intine, of course. This is the intine, this is the exine, triporate. A pollen tube is formed, and this pollen tube, it starts moving inside the style to reach the ovule. And when it is moving, suppose I am showing the pollen tube here, what happens? Very important, listen carefully. The nucleus which was in the tube cell, it is here. Okay, and what happens to this generative cell? Generative cell nucleus also divides and it gives rise to the male gametes. So there are 2 male gametes. 2 male gametes, and this will degenerate. The nucleus of the vegetative cell, it will degenerate. So finally 2 male gametes are formed at the end of the pollen tube or when it is innervating there. In the next lecture, we will see what is the structure of anatropous ovule. Thank you.